Howdy, everyone. You're listening to Blue Jeans and Boots, where genetics grad students talk about how to navigate a PhD program while trying to stay sane. We also invite faculty from our interdisciplinary program here at Texas A&M onto the show and pick their brains about their research, what it's like being a principal investigator, and their journey to becoming a professor here at A&M, or lots of other places. In this episode, we'll be talking to Dr. Anne-Ruxandra Carvunis. Dr. Carvunis is an assistant professor of computational and systems biology at the University of Pittsburgh School of Medicine. She earned her PhD in Bioinformatics from the University of Grenoble and was awarded the NIH Director's New Innovator Award in 2019. Welcome to the show, Dr. Carvunis.
Thank you. I'm very happy to be here.
We're excited to have you. Is this your first time in Texas?
My second time.
Your second time? When was your first?
2019.
Oh, really?
I think. There was a conference in Austin, Texas.
Oh, okay. Very cool. And you're here for a conference now.
I am.
Which I guess already happened. It's been the last 2-3 days?
Yeah, it started Monday, and this morning was the end of it.
Oh, wonderful. How did it go?
It was wonderful.
Good. Did you get to meet lots of interesting people, give a lot of talks? You were one of the big speakers, from what I understand.
I was one of the 2 keynote speakers.
Awesome.
It was an incredible honor.
Oh,
good.
Good.
And I did get to meet a lot of wonderful people.
Good. What was your talk about, can I ask?
My talk was about de novo gene birth, which was the topic of the conference, and focused on the concepts of what is a gene, what is a function for a gene, and where does this all come from? Right.
Those are some big, basic questions that I feel like even as a geneticist, I don't think about that often. But it's an important thing to know what a gene is and what a gene function is, like how to describe something like that.
Absolutely. And those are topics that don't get discussed much at school, but in fact are widely debated. And the Wikipedia page for the word gene gives a good introduction into this. And you soon discover that there are pages and pages and pages of literature and fierce debates as to what those words actually mean. It turns out that during our education, we kind of get siloed without realizing it, just because of what country we're from, what specialization we take. Wow. And then as soon as you start doing interdisciplinary research, you realize that other scientists have a completely different understanding of what these words mean. And this understanding is conceptual, of course, but translates to extremely different research practices and practical lenses, experimental and analytical, into how the elements are studied.
Wow.
And that leads to a lot of misunderstandings among scientists, misinterpretation of papers from slightly different disciplines. And that's fascinating and complex. There is no right answer, but I hope that by looking into this and thinking about what we actually mean, it helps refine our research questions and understand our colleagues' work better.
Yeah, no, that's like a very serious communication issue, almost like at the core of genetics, is if we don't agree on what a gene is, how are we supposed to agree on anything about what that gene is doing or its role, especially conserved genes across different organisms? Like there are things that in theory we have, plants have, fungi have, and bacteria have. And so how are we supposed to have conversations about all of that if we don't agree on what it is we're talking about in the first place?
Well, certainly. And the problem becomes way worse when you think about, like I do and the people at that conference do, new genes.
Oh, that's a good point.
Because in the context of emergence, of novelty, things become much murkier. When you refer to conserved genes, these are actually the ones for which there is a least debate, because it's very simple in some ways. Oh, this is a piece of DNA that encodes a product, a protein, that exists for a historical reason to mediate a molecular activities that is essential for life. That is why it is still here after so many millions or billions of years. But in the context of emergence, when something is new, it's by definition not yet required for life, since life existed before it did. So that's out the window. For instance, we also, with new discoveries in genomics, realize that lots of pieces of DNA are expressed. They make transcripts. They make proteins, typically microproteins, very small, that are just being discovered now. And we don't know if they do anything or what they might be doing, but we do see that they physically interact and participate in cellular systems. But is that important? Does it matter?
And are they genes?
Are they genes?
That's the $1 million question. Where do you think, in education, like up until we started talking just now, I thought I was pretty sure about what a gene was. Do you think it's just, I guess like you said, it's different disciplines define it in different ways for the most part? Or do you think there's other things at play about how different people look at what a gene is or isn't?
Both, as well as different periods of time.
Oh, okay.
Because our understanding evolves with more data.
Right.
The word gene exists since way before we know about DNA. The definition, the original definition of the word gene is a unit of heredity.
Oh, that's right. And people thought it was proteins for a little while too, right? There was like a period of time where before DNA it was
Gregor Mendel didn't know,
That's true.
It was however characters, phenotypes are encoded.
Yeah.
And so then, like with the advent of, molecular biology, then for a while we saw genes encoded enzymes.
Right.
And that's it.
That's right.
And so you see what I mean? And then like year after year, then people realize, but there's splicing. Oh, so can be multiple proteins for the same gene. And then people realize that, I'm not going in order, but another example is, the many traits are not driven by a single locus.
Right.
They are polygenic. There is a whole network of alleles and of molecules that drive a trait, a complex trait. So what's the unit of heredity? So it's between, the history, the subdiscipline, the country really, because different countries have had scientists and professors very influential at different times.
Right.
That have influenced the textbooks of that country, for example.
Yeah.
So it keeps going on. I highly recommend you check out the Wikipedia gene page as a start.
Incredible. I've never had anybody on the show who has been like, look this up on Wikipedia immediately. It'll give you great insight.
I can give another suggestion since some of the students listening and you are into genetics. Have you ever heard of the ENCODE project?
Oh, maybe in passing. Could you describe it a little bit?
Right, yes. So after the human genome was sequenced in 2001, big efforts went on to try to find the genes and then try to find the functional elements more generally of the genome. And one of these big efforts in the United States and around the world was called ENCODE. And it was a giant consortium of many labs that worked together collaboratively and did all sorts of characterization of the genome, the human genome, such as RNA sequencing, ChIP-seq, histone, replication, origin, so many different ways to look systematically and upload data in a format that was compatible so that we can look at what is going on the genome. And in 2012, they published a summary of their result. And some of the authors of that work, as well as journalists who just talked about it because it was so great, concluded, oh my God, amazing, the human genome, 80% of it is functional. And that was the start of what has now become known as the ENCODE controversy.
Oh, wow.
Oh, yes.
They started a scandal.
Scandal. Because, in particular, one type of biologists were outraged at this claim. And it was the evolutionary biologists. Because between 100 years of theory and a lot of analysis of comparing the genomes between species and among individuals, they had concluded that only a very small fraction, a few percent of the human genome was under evolutionary constraints.
Oh, okay.
And therefore, it's not compatible.
Yeah,
that
the two
80%
collide.
Is functional or 3% is functional.
Mm-hmm.
One scientist in particular, Dr. Grauer, published a piece in 2013 whose title is a bit long, so I hope I don't butcher it, but it's something like on the immortality of television sets. What's the rest of the title? Something about the evolution-free gospel of ENCODE.
Oh, wow.
And I actually recommend reading this piece, although I will give you a caveat. While entertaining and making strong points, the piece is pretty rude.
Oh, wow.
But it makes a point. And it makes that point that function is a historical concept. Mm-hmm. Function refers to the reason why an organ, a trait, a locus, a characteristic exists and is being maintained by selection over time. And they argue that a polymerase randomly transcribes some meaningless RNA at some point doesn't mean that this region of the genome is functional.
Right.
And this controversy just went on and about what is function in biology and in molecular biology in particular. And that yielded so many thought pieces, so many pages written on this. And one of the most thoughtful in my mind was written by several of the ENCODE members. The first author is Dr. Manolis Kelis in PNAS 2014, if I recall. And it is about how we all have different lenses into function. And they draw a figure that's very nice. Like if you imagine the human genome as a big circle, then there's a piece of this circle that's empty. And then there's but a big piece is like light blue for what ENCODE finds as low activity. And then inside it, a bit darker blue for medium activity. Even darker blue for more high activity. And then on top of that, there's little circles for mediates of phenotype based on genetic evidence, experimental or from human variation. There is a protein coding. There is biochemical evidence. If you take all of that, those circles, they barely overlap. And why is that? It's because we only have empirical lenses, different things that we can measure. And they don't tell us the same thing.
Right.
But they're all true. So it's very interesting. And so this is, I think, great for undergraduates to read, to those two papers, one after the next, to see how there can be different points of view and the importance of the concepts and of how what our concepts really mean from a pragmatic sense and what we can and cannot infer from what we measure.
Right.
That's fascinating. And so I enter so many more, like so many trees were cut to talk about this. And not just in science, right? In philosophy, in history, not just in genetics, in physiology, in taxonomy, like that. Those are very, like a lot of people think and write about this all the time. And so did I, together with a philosopher and a rhetoric scholar and a biochemist.
Oh, wow.
We all got together to understand how are people in the field of de novo gene emergence, the concept being the birth of new genes from previously non-genic regions, how are people using the word function?
Okay.
And we found that everybody uses it to mean something completely different. And then when we read the same sentence from the same paper abstract, we understand completely different things.
That's crazy. That is insane to think about.
Yeah, it's insane to think about, to realize, but it's a fact. And so this is my advice to the students listening. You can use whatever word you want, but explain what you mean to try to maximize the chances that your reader will follow your thoughts.
Yeah. Wow.
You didn't expect that, did you?
That blew my mind. Well, and I'm trained as a molecular biologist and a botanist, and I know the debate about whether or not something is a new species.
Yes.
And I never realized how much it would parallel whether or not something is a gene or whether or not something is functional. It really depends on how you define a gene, functional or species in this scenario. And it's hard, and people still debate about it.
Yep. It's very funny. It's a bit like colors. It's very easy to say, this is black, this is white. But where does it stop?
Right. Yeah,
there is a whole shade of gray. So the subtlety is at the border.
Yeah.
And that's where the interesting
stuff
matter lies. Yes.
Wow. We've talked about a lot of complex systems, and I know that you study complex systems sort of as a general in your lab. Can you talk a little bit about what your lab focuses on specifically, your research group?
Yeah, we want to understand the mechanisms of change and innovation in biological systems broadly, but with a focus on molecular systems. So those big, beautiful, complex and dynamic networks of molecules that interact with each other in crazy, complicated way, but that somehow integrate themselves into making life happen.
Right.
And first of all, how are these systems organized? And second, how do they evolve and innovate? So what are the roots of novel phenotypes, novel characteristics? How do these systems adapt to challenges in the environment, etc.? And in this context, our main focus is the birth of new genes. How do thousands and thousands and thousands of elements that we call the protogenes, which are completely new, like we discovered, like our field is discovering them as we speak, but they are translated elements, very small, completely species specific, and they outnumber the genes that you know and love and are annotated in the genome of your species. How do they integrate with the cellular network? Are they modulating pathways? Are they contributing to making each species unique? And how? These are the questions that we do not have answers for, but we are working towards.
That's so much. It's all so complicated, and there's so many elements that goes into everything. But it's interesting because the more complex a system gets, it's almost like the clearer it gets at the same time. And just in the sense that, like you said, that some of the elements might be, for example, species specific. I still have the botany going in the back of my head. Like that would be helpful to people to know what makes one thing different from another thing, even if on paper they look very similar. If there's something that we can assign molecularly that's different about the two, it gives us more freedom to say, okay, this is X and this is Y, and we can say how and why they are, which is kind of insane to think about. Somehow the more complex you get, the simpler the bigger picture can become a little bit.
I appreciate your optimism.
Do you have anything going on in the lab right now that you're really excited about in particular? Without, spilling all your secrets.
I cannot choose my favorite baby.
I understand that was an unfair question for me to ask. What are some examples of some, biological networks that are really critical to things that we learn about in school? Like people will learn about like respiration or photosynthesis or things like that. Are there more complexities to those sort of basic systems we learn about than we even realize and we're taught about in school?
Of course. Mm-hmm. That goes without saying. But perhaps what's most exciting to me is how all these systems are actually connected with each other.
Oh, wow.
Right?
Yeah.
How signaling, for instance, or physical separation inside the cell are dynamic and modulate how different subsystems are active relative to each other, compete or coordinate.
Oh, wow.
Mm-hmm.
See, it's complicated in pieces, then it's complicated all together. So the whole thing functions together.
Yeah.
As a whole.
Right.
That's insane.
I think that's what I think is really cool.
Yeah, that they all somehow work together. Mm-hmm. And do you look at all into like the origins of life itself? Because I feel like you're getting closer to like what makes life exist if you're looking at like the nuances of what changes between different parts of life.
I like to sometimes think about this, but I don't work on it. Okay. The time scales I look at, while very ancient for me, like millions of years, there's a blink of an eye for the history of life.
Yeah.
There nothing that I study mechanistically can apply to origins of life itself, because everything I study happens within a cell. So it's post-existence of a cell. And mostly also post-evolution of sexual reproduction and things like that, which are relatively late events in the history of life.
Yeah.
Perhaps some of the principles of what makes a novel protein, how does molecular adaptation and coevolution take place, perhaps some of those principles might be helpful for my colleagues interested in origin of life. But my research cannot be directly applied to pre-cell life.
Okay. Yeah, that makes sense. Curiosity question.
Right, yeah. That's a great question.
What about, origin of life aside, do you know where do genes come from? Do you have like a working hypothesis about where they start? Obviously, we talked about protogenes a little bit. Could you talk a little bit more about that idea?
Yeah, absolutely. So it's actually, my hypothesis that I work on is actually pretty simple. In the genome, the canonical genes, like textbook, are discrete loci scattered along the chromosome, and each of them encodes a functional product. In between the genes is what we might, for simplicity, refer to as non-genic region, also known as intergenic DNA, non-coding DNA, junk DNA, the dark matter of the genome.
She's getting more excited visibly as she's saying this. For all the people that are not watching the video right now, I just want everyone to know that the progression is fantastic.
Yeah, I'm passionate.
The dark matter of the genome.
In the mid-2000s, were the development of the first technologies that allowed to look at RNA expression at the genome scale. First, oligotiling arrays, and second, RNA sequencing. And the major surprise from these advances was that a large fraction of the genome, including the non-genic regions, were transcribed pervasively. And I, for independent reasons, was wondering where genes come from. And I also was learning, I was a graduate student, what does a genome look like? And it's very obvious. We'll see immediately that a genome in the intergenic region has a lot of ATCGs, and that therefore the probability to have an ATG or a stop codon is extremely high, and you can even calculate it mathematically given the fraction of A, T, Cs, and G.
That makes sense, yeah.
Yes. And I thought, but if the genome is pervasively transcribed into RNA, why would the ribosome not read those transcripts? And I thought perhaps there is translation of what I called non-genic open reading frames. So, and yes, that's what happens. This is what I and then many others have shown. And this is what we call the protogenes. Just the products of pervasive expression of regions that are, we thought were non-coding and are intergenic, non-genic. And those regions, unlike most of the genes, the coding genes, which, like you said, tend to be conserved for millions of years, plants, animals, fungi, the non-genic regions are not.
Right.
They evolve really, really fast because they are not under constraint. So as a result, when those fast evolving DNA regions happen to get translated, the little putative peptide proteins that come out of it are completely new. Nature has never seen them before.
Right.
Neither has your cell. And so what happens? So I originally hypothesized that most of them would be insignificant, but perhaps sometimes some of them would be beneficial.
Right.
And just generate novelty. And that's where genes come from.
Awesome. That was a great answer.
And of course, that was 10 years ago. So now we have more research, more data that supports and especially refines this model. And one of the most striking things that has come out in the past couple of years and was very evident at the conference that just ended is that even those little protogenes that you would think are random, they are not quite random.
That was going to be my next question, was how random are the protogenes and how random is it that they get picked up by a ribosome and how random is it that they turn into anything? Okay, so not quite as random as you would think.
Not quite, but we don't understand.
It's going to be my next question. It's like, how do we know?
So you come and study this. And strikingly, so it seems like their expression is regulated.
Okay.
We don't know how. I mean, we start to observe, but we don't know how. We don't know how that changes. We see some biases in the genomic locations that make protogenes. But we don't know if that is condition. We think this might be condition dependent, perhaps, and this is wild speculation. Students, do not believe what I say, just imagine with me. Perhaps in certain conditions, the cell will actually produce a lot of transmembrane proteins that will go and modify what's in our lipid bilayers. Perhaps in other conditions, it will want to make a lot of secreted proteins or a lot of disordered, flexible,
Yeah.
So I don't know that, but this is possible.
Yeah. It could be a condition specific,
It could
scenarios.
It could be a constant adaptation of always some novelty, but of different types.
Oh, wow.
What we do see, which really surprised me, is that at least a substantial fraction, I cannot know the actual fraction, but there is a fraction, not null, of these elements that despite being entirely novel, just appeared. If you genetically manipulate them to inhibit their expression, it leads to observable changes in the behavior of the cell or the organism. Yeah.
Mic drop. I was not expecting you to say that either. I'm surprised this episode. I'm having a great time.
Well, I'm surprised by our results as well. I think it's fascinating.
So maybe the protogenes aren't necessary, we talk about necessary and sufficient in school. At least that's a lot of what I was taught about genetics is genes are necessary or sufficient, or ideally both. And then you can test that by knocking things out. But if you knock these out, it's not necessarily that they're necessary, but they're sufficient to perform a task that they're suddenly not performing, and it leads to observable changes. Have you seen this yourself, like in your research? What organism, can I ask, that were you working with?
You can ask, yes.
Okay,
That's
curious.
what I'm here for. In my research, I observed this with yeast.
Oh, cool.
Yeah. And in this conference, I have heard parallel discoveries. This is all very recent in other organisms as well.
Oh, that's huge.
Yeah.
Oh, cool. Are any of them multicellular? I just curiosity. Oh, okay, cool. Oh, that's exciting. We're really at the forefront of knowledge here too. That's
absolutely.
insane.
Do not be surprised if, in 2 or 3 years, it turns out it was all an artifact. This is how science works. But right now, we're all very excited.
Right now, it's fun.
Yeah.
Would you say that whether or not something... I'm trying to think of a good way to phrase this. I guess, what would you say is the biggest challenge facing the field right now of systems biology and of evolutionary systems biology, I guess?
There are so many challenges.
That was a big question.
The field of evolutionary and systems biology. I think it's about measurements.
Okay.
So the technologies are really improving and new technologies are invented all the time. But if we need to understand, we need to model. So we need data that is reliable, that is complete, that, that we can base our observations on, like naturalists. And I think that is a big challenge.
Yeah, that makes sense. Whew. That was a lot of big topics, big complex discussions. So I'm going to scale it back just a little bit and ask you just about your journey to becoming the professor that you are today. What did you study when you were in graduate school? What got you interested in the field?
Yeah. So I did my undergraduate studies in Paris in biology, biochemistry. I destined myself to study neuroscience, molecular neuroscience. Did a master's in neuroscience and realized I hated it.
That'll happen.
Right. The difference between the topics that are fascinating in class and the daily life of research.
I understand completely. Right? Yes.
So I recommend everybody does a lot of internships to figure out what you don't like.
It's equally as important to know what you don't like as what you do like.
Indeed. Having figured that out, I almost quit science.
Oh, wow.
But a professor said, no, come on, I'm starting a new master's degree. So I joined that master's degree in interdisciplinary approaches to life sciences.
Ooh.
And that's where I discovered coding.
Oh, okay.
Which I fell in love with. I loved it. But then I did not get a PhD fellowship in my home country.
Okay.
Because I was trained as a biologist and I wanted to work in more of a CS or physics labs. And so nobody wanted to fund me. It was quite a challenging time.
Yeah, understandably.
And adventures later, I found myself in America. And a kind soul directed me to a lab that was doing systems biology. And that, together with the master's degree I had done before, really, really resonated with me because my passion for the brain from a few years back was really about complexity, about complex systems. How do matter that is interconnected serve to make us who we are and in interaction with our environment, and through reading a few papers and then discovering this lab in systems biology, I realized it's the same inside every cell.
Mm-hmm.
So I joined that lab in systems biology, and that lab was, and still is, the big challenge is to map protein-protein interactions at the genome scale using various high-throughput technologies. Fascinating stuff. And at that time, so I went and did that, and I loved it. And at that time, though, just a few years before I started my PhD, something happened, which was the beginning of comparative genomics in eukaryotes. Because the first eukaryotic genome was the yeast genome, sequenced in 1996. And then starting in 2003, people sequenced more yeast genomes of different species of yeast. And when they did that, they realized that some of the genes that they had guessed were in the first yeast, Saccharomyces cerevisiae, did not exist in the other yeast. And so they thought, oopsie, we were wrong. These were not genes, since every gene has to be conserved.
Oh boy.
Yep, you see where I'm going. But I didn't get there immediately. Mm-hmm. How I got there was because my lab and another graduate student who was there before, Chen Wu Li, they thought, oh no, our field has been mapping protein interactions with these elements and they were never genes. We must have a lot of false positives. So we looked into it and no, they behave perfectly fine.
They were real, yeah.
And I was naive. And I was like, but I don't understand. These are different species. Why can't they have different genes?
Oh, wow.
Where do genes come from? And
oh my god.
That's how I got there.
And that was how it all started.
Exactly.
That's a great story to tell. Oh my gosh. That is wild. And it's just crazy to think too that all of the comparative genomics started with yeast, and they looked at it and they were like, they can't all be genes. I can't imagine doing that today. That's crazy. Oh.
Yeah.
Wow. And that really answers my follow-up question, which was then, how did you get to where you are today? Which, I mean, that pretty much is how.
Yeah, exactly.
Where do genes come from?
Yeah. And one important note is that the scientists who rejected those genes because they were not conserved, they were very smart and they had good reasons to do that. And the good reason, the number one, is that they never really knew for sure what the genes were. Nobody does. If I give you now a file of ATCGs and I ask you to read it and tell me what it is, you cannot know. Mm-mm. Nobody can know. The best way we have, basically, is using comparative approaches, like what have we learned from the other genomes to infer what this one might be. Mm-hmm. The only way you can see that this is like a horse genome is if it looks like the genome of a horse.
Right. Yeah. No, that makes sense.
You know?
Yeah.
So at the time, in the mid-90s, when they were sequencing the first eukaryotic genome, they saw that there was all these ATCGs and these start codons and stop codons, exactly as expected mathematically, except that there was a giant tail of longer open reading frames between start and stop codon. And that made a lot of sense because those they thought those must code for proteins. And also they happened to be big enough to code for what we at the time knew were proteins, meaning folded proteins. So they thought, and they already knew a bunch of proteins. So from what they knew and the sequence that they were discovering, they thought, all right, let's just say, as a working hypothesis, they were never sure, they never pretended to be sure. Every time that we have 300 nucleotides without a stop codon, we're gonna hypothesize that this must be a gene. But they knew it wasn't perfect. Right. So when they sequence, the community sequence other yeast, and sometimes those 300 nucleotides or 350 nucleotides in the other species, there were stop codons, it wasn't. It was very natural to think, it's natural, like some of our first guesses were wrong. Right. You know what I mean?
Yeah.
So it all stems from very rigorous and good science.
Yeah.
But also takes like some naive students, like myself at the time, to question Yeah.
What is that?
So that's how I got in there. And of course, when I did, I found that there was a few papers suggesting the potential of new genes being born, quote unquote, from scratch. But really not a lot, because it was impossible in people's mind. There were a few pioneers that said, like, it's possible. We have one, we have two. But this really hadn't taken off because there was no understanding of a potential mechanism.
Ah, okay.
Right? And so that's where really I came in, starting from a naive question, educating myself, and being in the right place at the right time. Because I was in a systems biology lab. I was doing computational biology, so I was used to working with data and thinking big about the whole genome, the whole cell. And around the same time, like as I had already been working on this, like on the side of my protein interaction project with my PhD advisor's support, then something amazing happened. Is that a professor in California and his postdoc invented a new technology called ribosome profiling.
Okay. Which
consists in basically sequencing the little bits of RNA that are inside a ribosome. And so, like, when they did that, they published a great paper in 2009 in Golia et al. And they proudly said, yay, like, this technique works. You can look at translation now. And as proof that it works, look, 99% of our ribosome reads map to protein coding genes. So I was like, what about the 1%?
What's in the 1%? Wow. Talk about good timing.
Yeah.
That's crazy. Oh, wow.
Yeah.
Did you know at the time that like all of the research was sort of falling into place and your interests and what was going on in the field were sort of aligning? Did you know you wanted to be a professor at that point and have your own? No.
I wasn't even thinking about it. But I also didn't know that I did not want to be a professor. I just wasn't thinking too much about the future. I was thinking about science.
Oh, okay. Yeah. Awesome. That's a great answer. I'm always curious, like people usually have a like, oh, I lean towards industry. Oh, I lean towards academia. You were just like, I was thinking about science and everything else was secondary.
Pretty much.
Awesome. What kind of advice, I guess, then would you give to graduate students who are in grad school now maybe thinking or maybe not thinking about what they want to do in the future? If you could give them like a piece of wisdom.
I'd say if you know what you want to do, go for it.
Okay.
If you don't know what you want to do, that's totally fine. Yeah. You'll figure it out. Go where it's fun and where the doors open and there's light. Follow your heart.
Yeah, I love that. That's good advice. Yeah. And I mean, we already talked about how important it is to know what you like and what you don't like too, which is also, I think, very true and something that we don't talk about a whole lot in science. But if there's something that doesn't resonate with you, you don't have to keep doing it.
Exactly.
You can move on to something else. And obviously you did, and now you're
a
pretty
happy.
Big deal.
I'm not saying it was easy, right? So like I think we can make maybe a distinction between what we don't like and what we don't like. There's kind of two things. In my case, there were some aspects of neuroscience, but also other, because I did lots of internships, like that I just really, like, I could not stand, for example, waking up and killing a rat. That was just, this was not possible for me. But when I did my PhD in bioinformatics in this systems biology lab, there were also a lot of aspects that were hard, that I found boring, that I thought too challenging, too hard. And I'm really glad I didn't quit.
Yeah.
Right? So I think I wanted to many times because I thought, it's too hard. I want to be on vacation all the time. I want to make more money.
All the graduate students.
Right. Those are valid feelings.
Yeah, of course.
But I'm just really glad I persevered, not only because now at my age that we shall not say, I'm very happy, but also because it showed me what I'm capable of. And I would have never known if I hadn't gone through those times of hardships. I would have never known what I'm actually capable of. So I'm glad for the adversity. And I tell you, graduate students, keep trying, and be resilient, and your older self will thank you for it.
That's excellent advice. And what about for undergrads? Obviously, they have their reading to do. Look up gene on Wikipedia. And what was the article after the ENCODE project?
The television sets.
Oh, the television sets, right. Okay, so we've got reading assignments for everybody. What would you tell undergrads who are maybe thinking about going to graduate school or thinking about starting research?
To try and do as many internships as possible. Go and get experience for so many reasons. First, because not two labs are the same.
That's true.
There's so much diversity across institutions, within the same institutions, within different departments, different labs in the same departments. There's so many different aspects of research life, and some will matter to you and some will not. There's really too much to discover, to experience, both scientifically in the ways of working, the ways of interacting with colleagues, with PIs, the types of systems, experimental or computational, the types of questions, the types of articles. So this is my advice, is to go and explore so that you learn really, you broaden your mind. You might discover something that you would have never thought be of interest. I didn't like evolution in undergrad.
Really?
I mean, I didn't really care. I thought it was for hippies. To be honest, I wanted to do something serious.
Awesome.
You know?
Yeah, I understand.
I skipped. I think my teachers retired, so I can say I skipped every statistics and computational class. I thought that was boring. I was like, oh, p-value, probability, who cares? It happens or it doesn't. I want to study animal behavior. Look at me now.
But here you are.
So I think it's very important to broaden your mind. You never know where the passion will strike.
That's true.
And also, it looks really good on your applications.
It does look really good on your applications to do lots of different things and to try it out. And I know there's some people that'll preach against the diversity of experiences. They're like, you need to look like you have one path that you take. But I don't think that that's true at all. As someone who also had a diversity of experiences in undergrad, I think it looks good to do lots of different things. So I'm going to second that. Try everything, because what do you have to lose also?
Yeah, and also if you fall in love, like, you can stick with it.
That's true.
Then you have one path and that's your passion, and that's great.
Yeah. And then you found it.
Yeah, exactly.
Awesome. With that, is there anything else that you want to leave our audience with before I wrap things up?
Thanks for listening.
Yeah, of course. Thanks for coming on the show. We really had a great time talking to you today. Today's episode was hosted by me, Serina DeSalvio, and produced by Aaron DeSalvio. This podcast is brought to you by the Texas A&M University Interdisciplinary Graduate Program in Genetics and Genomics. If you have any questions about our program here at A&M, please feel free to email us at bluejeansandboots@gmail.com. You can also find us on Facebook, Instagram, Twitter, and TikTok at Genetics Podcast. Thank you for listening, and we'll see you next time.